You can use this video to fill in the second to last page of your notes packet, the one that goes over dehydration synthesis and hydrolysis. So here we're looking at question number 33. The reaction that joins two amino acids to form proteins is called dehydration synthesis. And the bond that's formed is called a peptide bond. Therefore, two amino acids linked together is called a dipeptide, because it's two amino acids joined by a peptide bond. Now, as you start to draw the pictures, you're going to draw what I'm showing you. Feel free to stop it, pause it, draw what's here, and then resume the video whenever you need, whenever you catch back up. Okay, so you have the ability to use that pause button. So the first thing I'm going to do is draw an amino acid. I'm not going to draw out the R group. I'm just going to label it as R. So here's my nitrogen, my amino group, that's my NH2, my central carbon with an H. There's my R group. That could be a CH3, it could be an H, it could be a big ring structure, it could be lots of different things. And then I've got my carboxylic acid here. Okay? And there's my first amino acid. Now that's going to get joined to a second amino acid. There's the R group, carboxylic acid. Okay? When those two get joined together, what will happen is that this OH group here and this H group are going to have to come out of the reaction so that we can form a bond between this carbon and this nitrogen. Okay? And in order to form that bond, you've got to take out that H and the OH. So when these two things join together, that's going to form H2O, or water. Okay? So you're drawing what's in purple, and you're then going to draw what the actual dipeptide would look like. So you're going to have nitrogen with two hydrogens, central carbon, hydrogen, there's the R group, the carbon with a double bonded oxygen. Now here is going to be the peptide bond, and that's going to be linked to the second nitrogen. Hydrogen, carbon, R group, OH right there. So my peptide bond is this. And the other thing that I'm missing is a water molecule. So every time you form that peptide bond, a water molecule gets released because of the OH right here and the hydrogen that have to come off to be able to form that bond. Those are going to come off and form this water molecule. Okay? So that's our example of dehydration synthesis of two amino acids. Now we're going to move on to carbohydrates and show how that. Okay, so here's a glucose molecule. Go ahead and draw that. You can see the blue hexagon. Pause the video if you need to be able to draw that, and then we're going to add another glucose molecule too. Alright, so there it is. And if we're going to link those two together, we have to be able to take out parts to be able to form an actual bond. So this hydrogen is going to come off, the oxygen next to it will stay there, and this OH group is going to come off. Those will go together and form water. Okay, and the carbon will then get attached to the oxygen. So let's draw that. Feel free to pause the video and draw it. So there's your two carbohydrates joined together. Okay, and that's going to be what it will look like using that same type of reaction. So that's number 34. Now, when we go down to number 35, this is sort of the opposite. Now we're going to do hydrolysis. So let's say we have a big long chain of amino acids that is a protein, and or maybe it's just a dipeptide that's stuck together. So we're going to draw that dipeptide and then show how hydrolysis would work. Okay, so there it is. There's our dipeptide. Feel free to pause the video and draw it if you need to, and then resume. Don't forget you've got this dipeptide here, and then you need water to be able to fill in the spots where the bond is going to break. Right here is your peptide bond. Okay, so that's going to break apart. Here's your water molecule over here, okay, that I've drawn. That'll break here, so you get an OH group and you get an H. And they will go right into the spot where the peptide bond is. All right? So I'm going to draw those H and the OH in yellow when I redraw the new amino acids that are separated so you can see them. So this one will go over here, and this one will go over here. All right, so draw that. And you notice this OH group that refilled that section of the COOH group, that is coming from this OH in the water that we took apart. Okay, something had to rebuild that bonding site. And this H right here that completes that amino group in the other amino acid, that is coming from the H that was in the water. And so this, if you had a big long chain, everywhere you split one up, or split between two amino acids, you would have a water molecule that would have to be used. Now remember, the R group that you see here does not mean it's necessarily, there's no R there. I'll show you what those would look like. So if I was to replace the R groups with actual R groups that you might find, this is what it would look like. They would look just like this when they were up here in the original molecule, and they would stay the same as they are. Because remember, each amino acid is defined by what its R group is, and there's 20 different R groups, therefore 20 different amino acids. What we're really focused on when we're doing hydrolysis and dehydration synthesis is this peptide bond that forms or breaks between the carboxyl group of one amino acid and the amino group of the other one. We will practice this and use this in class, so make sure you get it all drawn out.